beautiful stills like this. Can you hear me? Yeah. All right. So, and basically, I have to say I'm working with insects, with fruit fly. So, I mean, if you expect like amazing mammalian brains or human brains, I mean, no, this is not about that. So what I'm dealing with, what do I want to do with Blender? Basically, what I have is this stacked sequences of images. These images are optical sections of samples. So in this image, I have all the information that is describing my objects labeled in the sample. And now I want to extract the 3D from them. So basically, there are many ways to do this, but they have some problems, most of them. And so let's get into this. This kind of data that I have has to be what we call, in science, we call it segmentation. So if you have just an image that has a lot of objects in it, you basically want to define which parts of the image are the contour of your 3D object. And so there are several approaches that mathematicians just love to use, which are automated, and they're based on the pixels in the images. And well, I don't know if some of you may be familiar with the VTK, the Visual Toolkit, and also with Amira, and also volume viewers for various applications. But these have a lot of problems. And the main points I can make is that if your images are horrible, you're going to get horrible 3D models. So segmentation cannot automatically detect what your eye can see. And then it's also very, very intolerant to whether the items you want to segment in your images are not uniformly labeled. So the label is kind of dimmer in some sections and it's not stronger in others. Then, of course, if you have many objects and they're all labeled the same way, so you have different parts of this brain, different cells, different tissues, and they're all the same color, even though you can tell them apart, the machine will not tell them apart. So then what you end up getting is something like this, which is, yeah, wow, beautiful. I mean, this is bullshit. Honestly, I mean, from a mathematical point of view, it's such an interesting exercise to create this 3D, but it's not usable. Because in here, I cannot tell each individual fiber. I cannot tell each individual object. There's just no way, I mean, at least nowadays. So what we're left with is, since we cannot automatically generate these nice meshes, is to go to manual segmentation, which is a pain. but I mean, it's slow because you have to basically go over these stacked images that represent your sample and highlight in whatever way you can the regions of your image that make up your object. And so, of course, it's user dependent. I mean, everybody has a different eye. Everybody will see different things in the same images. But it's not so bad. It's not so bad. We can say, I mean, we have to answer this question, basically. So what's this model to be for? And I'll try to answer that. So what I have is basically, I created this program in ImageJ, which is this very nice public domain application for handling images and image analysis. It has like literally about 1,000 plugins for all open source GPL to do whatever you want. It's Java based. And so what I can do basically is load in here as many images as I want, and then I created these tools to just create, for instance, Bezier contours of different aspects of my images. These images are layers of the sample, so they represent a volume. And so then, of course, I can just quickly, have a look at how these contours look like. And I'm left with the problem of making a 3D out of all these contours, which is also an old problem of triangulation and making up, some nice skin out of it. So basically, this is what I want. Go from there to there. And this looks quite simple to the 3D community, maybe, but it's just not. I could not find a single program out there that would do this transformation from just plain curves in the 3D space to a skin without failing. Because there are many occasions in which I can show in a moment, but the curves are so different from one section to the next that the skinning procedure is just horrible. It just creates a horrible skin. So I can show this. Like, for instance, this is what Maya can do. It just looks pitiful. What is this? Is a process of lofting. It's getting all these Bezier curves and trying to match them with the next, the following Bezier curve, and trying to make a skin. And of course, I mean, in whatever way it does, it may be mathematically correct, but it's just not proper. And then it just do some plain old triangulation and trying to match points. It's also not proper. It's failing to recognize that what we're trying to match, I mean, these two profiles, which are so different from one to another, they just cannot be trivially matched. So for these, I came up with this mathematical paper from like 1974. I mean, these guys were just trying to put together strings of characters in a sentence, trying to find how two words diverge one from another. So they have these sequences of letters. They're trying to detect which letters are missing, which letters are inserted. And some guys said, well, we can do this also for vectors. And then we can represent any Bezier curve as just a string of points, which these points are just vectors pointing one to another. So that's what you have up there. So any curve is just a sequence of vectors. And so now by applying this algorithm, which is basically a super simple two-dimensional matrix that just tries to compare one vector with the other, well, you can detect that there are parts that are missing, like the green part of the curve here. And you can detect the parts that have been inserted. And then you can create weighted curves in between the two that will then make a very smooth transition. And so I implemented this in Blender in a Python plugin in C, just for keycast speed, and that's what I can get. I can get just a very nice transition from one curve to another inside Blender. So the whole thing, just, I mean, in this case, this is just like 50 outlines of an MRI scan. So you can immediately get just by interpolating and so on using this algorithm, which is, as I said, is a Python plugin for Blender, just the whole skin. And as you can see how it works, it basically, whenever it detects that there are some inserted points, so it puts them, or it just deletes them if there are too many, and tries to find the best way to create a mesh in here. And so, well, just to have a look at this, what you can control in this curves is how many curves you want, and of course the resampling, so how accurate, how low poly your mesh you want it to be, or how high poly you want it to be, just because of the algorithm lets you do that, since you have to resample the curves. So that's how this crappy interface I came up with for Blender that I'm sure could have some nice reworking. And also, of course, let's not forget to mention, I mean, this is available, there's all GPL. I mean, my lab is just hosting this in this web page. And so, well, let's move on. So much for now for meshing, which already solves part of my problem. I can just do manual outlines on my stack of images and get the mesh. But then sometimes this is so slow that it's not worth it. Particularly if we have to simulate, for instance, tubes that have to go through my sample, through the sequence of images. And so what I cook it up, which is also, it started as a crude hack, and I finally kind of like it. It's basically, well, we can define a Bezier curve that goes over the 3D space of my sample. And in each point of this Bezier curve, I can define a radius, all right? So basically, this is what I have. I can draw in my own GUI in ImageJ, just a tube, and this tube will have a radius, and we'll have two control points for the Bezier curve. And I can set up some visual cues, so I know whether I am in the current slice, or this tube has been drawn in like previous slices, or in forthcoming slices in red, which is not very well contrasted here. But what this lets me do is very quickly follow these tubular structures, which in this case these are bundles of the neuronal projections of a lineage in the fly brain, but it can be applied to basically anything. And so very quickly I can get a 3D tube that represents this projection pattern. So in this case, I mean, I'm not going to go into details here because it's not but this is actually a fly brain, believe it or not. And so what we have is on the top, these are the two hemispheres. These are the eye fields, for instance. This is the optic nerve that goes to the actual eye. This is like the retina, like the occipital part in mammals. And so what I wanted to model here was some of the projections of these green cells out there all the way to the ventral nerve cord, to the medulla of this brain of the insect. And so just using tubes, I mean, in like 20 minutes, I can get a couple of tubes, and I can already instantly see if, like in Blender, just by visualizing it inside Blender, whether I have some overlaps on the projection patterns with some other marker I'm not going to go into now, but so I can identify the gene expression in all these different projections of neurons and see whether they overlap or not. So I can test for identity of these tubes with known publications, known descriptions of these projection patterns. And then of course I can extract beautiful renders that everybody envies in my presentations in the scientific community. So just to show some more examples, I mean, in this case, just using tubes and spheres, in like 10 minutes, I can just click here and there. I can get this beautiful render that shows me, in this case, this is some, wow, to summarize, there's an injection of a tracer that would follow up the neurons all the way to the cell body. So I can label neurons by injecting the ventral nerve cord, so I can find neurons that are projecting into the ventral nerve cord. And so that's what I get. But I can see now, mapping this with other renders I had before, I know which parts of the neuropil, which parts of the white substance of the brain, the fibers and the compartments, which parts they go into and which parts they actually don't go into, which parts they interact with. I mean, really quickly I can have a look at it just by tubes and spheres, and of course having a beautiful mesh rendered with this curve morphing module. So we didn't only apply this. I mean, our lab is working with a lot more stuff than just the fly brains. So just an example, this here is a worm, but it's a very nice worm that you can cut in two and will regrow its brain, for instance. So it's like kind of beautiful stuff to work with for biology. And so also using this approach of tubes and manually segmented meshes, and we very quickly generated this map of this invariant pattern of muscles in the dorsal skin and the ventral skin. Just some details. I mean, this is the anterior part. This is the head of the worm. And so in blue here is basically the brain, and in black you have the eyes. So, and these are all meshes, and just using the internal, Blender features that have been in Blender forever, and we just didn't know about. we can represent with transparencies these compartments in the brain, and which structures actually cross the brain and come out, the nerve roots, the commissures, and just very beautifully map the whole thing, which was not possible before. And so, and also, and this is my own crazy stuff late at night. I'm just trying to push the limits of Blender by saying, okay, how can I represent my images, my data inside Blender in a way that I can look at it. So in here, this is an outline of a neuropil. I mean, let's say this is where all the neurons are projecting to, and all these black dots are actually synapses. So where two neurons are interacting. And so the problem is this has like 125,000 little objects, and this can get really slow inside Blender. But so actually, if anyone has ideas on how to represent how many objects, that many objects inside Blender, so that we can.
Is Ton here? Yeah, like I don't know if it's true, but like in Blender, all the objects are stored as a linked list. So so to find an
a single object that I copy. If that's what you mean. There's only
yeah, but still, like the duplicates are I think objects on the object level, or are they? I'm not sure. Because well, anyway, like all the objects in a scene are in a linked list. And how many objects did you have?
Like 100,000.
Yeah, because then it needs to like iterate the list to find a specific one. And one, like, I don't know if it would help really, but we've been thinking of like perhaps using like hash maps, hash tables to sort of have an index of the objects and that sort of stuff.
I think over a code I would understand what you're saying.
Yeah.
Do you mind, I'm a biologist?
Yeah. But it's something that should be looked into, because it often comes with this sort of stuff. And yeah.
All right. Well, thanks. So hopefully in the near future we'll be able to just do all this crazy stuff to inside Blender, so we can see in 3D space which areas of the brain are actually contributing more to different projection patterns of neurons and so on. Sorry? The outline. No, actually the outliner is quite okay. Yeah, actually, I didn't mention so, but the outliner is so useful, so amazingly useful to just switch on and off objects, select them, and move them around. It's just, actually, I would love this outliner to be extended inside Blender if I can ask for that, just so I can right-click on it and edit more properties of these objects, transfer them to different layers, and so on. So just some two lines here. I mean, it would be really nice to have some way to access databases. I know from Python I can access my PostgreSQL databases with all my information. So then I could maybe visualize all that many objects, hundreds of thousands of objects, just by querying them, representing them, throwing them away, reloading them again. I mean, this has to be worked nicely, but I would definitely appreciate some comments from the source people. And also something I would love to have is standalones or games. I mean, so that I can have my 3D models, I can send them to some colleagues without asking them to learn how to use Blender. So, and VRML, I mean, don't get me started with VRML. But it would be nice to just be able to export this in some navigational system, just a camera and the ability to switch on and off objects. Just some super simple setup would be amazing for visualization of this kind of structures in biology. So just to end here, well, thank you to all these people, to this amazing program, ImageA. I suggest that you go and try it for image processing. It's about the best thing I've seen. Then, of course, Blender. This is my own cooked app, PostgreSQL based, that can handle like 1 terabyte of images without choking. And finally, all the institutions I'm working for at the moment. So, and thank you to you. Any questions?
Just a small one. If I understand, you make the segmentation manual now, not automatically. Is it right? So it's all the stuff is only for one data presentation, is right? If you have new data, then you have always to do the job for the segmentation manually. Is it right? Or do you have a semi-automated version?
I don't have a semi-automated one. I'm working on it. I have some guys in Zurich, actually. These are like real computer scientists who are trying to figure it out, at least to make it semi-automated. But it's not easy, because it depends so much on each sample. Each set of images has its own little pitfalls on how you can segment properly objects.
On these scientific images, they are always the same colors, or they're very different? Because in the GIMP project, they added some stuff about getting the borders of objects in images. I forget the name. It's GPL. There's an algorithm to get the border of when you have a person behind a blue background to just paint around it, and they draw the selection of the object.
I know of several algorithms to do this. The problem is none of them is reliable completely, because it depends so much on the images. If your object has fuzzy edges, blurred edges, then where do you define the limit? You have to say that your eye is saying so, or you're saying a parameter. And if this fuzzy edge changes through your sample, because as you go deeper in a sample, for instance, the image quality is worse. So then your object is very nice and clean at the top, and at the bottom it just looks like crap. So the same algorithm will fail. I mean, will be fine at the top and will fail at the bottom. So.
Did you try to use blobs from all the image slices? I use it blobs. It means these objects which metaballs. Yeah, I know that from Porphyry I used once. image, a cloud of points, of vector points, three-dimensional points, and I assigned metaballs to every of this point, and so they glue together and give a whole shape with it. Yes, that's you just said every point in the 3D space is a metaball, and so if you grow make them a little bit bigger, they match together, and so we have the whole shape. And
oh, I'm sorry. I just wanted to know how do you manage when you have, for example, two closed curves in one plane and one closed curve in the next plane? I mean, how can you merge different curves? Because I can understand that if I have a circle and then a different shape, it's okay. But what? Yeah, for example, like in lung, you know.
This is actually stuff I'm working on at right now, at the moment. But it's not too difficult, because with the same algorithm, you can detect which parts of the curves are similar to which contours in the next section. So then you can decide rules for saying, okay, this part of the curve will go and correspond to this profile in the next section, and this other half will go to the next one. And you can nicely do it. But this is I'm working on, and I need at least another month to finish it.
Just one question. Is it possible with your technique also to use this for interpolating video images for slow motion? I mean, if you have a series of images and you want steps in between, images in between. And so to do this interpolation, because sometimes you can just double the frames, but this would not give the transition between the frames. But yeah, and this has to be done by hand. Yeah. Mm-hmm. Oh, okay.
Well, I can still imagine some kind of semi-automatic thing like lasso tools. Yeah, and then so they suggest you what might be a good contour, but then you adapt them by hand, and also they suggest you what is similar to the next plane. So instead of saying this is what in the next plane, it's suggesting you. So, and you can then say yes or no. Yeah. I'm. Yeah.
Thanks.
Can you tell something about your image data? Are you using only the MMI images, or do you use multimodal images?
Well, is it working? No. Well, these sequences of images mostly are from confocal imaging. Confocal is this laser scanning microscope that has basically an iris, a pinhole. So the reflected light, you can control from which Z level in your section, in your sample, you're actually acquiring images from. But then I'm also working with EM. Like my current project is, EM is electron microscopy. Whoa. So with electron microscopy, you can also get serial sections. This is actually real plastic embedded 60 nanometer sections. And then you can outline a model from there.
There are similar problems when you do computer controlled radiology. I don't know how it's called, and don't know the English word, kernspin tomography. You know kernspin. Maybe someone can translate it. Kernspin tomography. You do sections of the body, very lots of sections. And yes, MRI. Okay, MRI. Aren't there similar problems in this area?
Well, keep in mind we're looking at the fly brain here, which is about 200 micrometers. And MRI resolution is about 1 millimeter. So we're well under the resolution of MRI, unfortunately.
Yeah, okay, but
the algorithms. The image processing techniques, yes, but how they are, most of these MRI scans, how they extract surfaces and so on, is in these dead, unusable models kind, that you have one big blob that is your object. And there's like no way, at least at the moment, to go and just segment parts of it. So we have another speaker. We should move on.